This is a production of Cornell University.
Thanks, Eva. Okay, thanks, Adam. Can everyone hear me all right? Okay, good. Thank you again for the invitation. I've had a wonderful time. It's my first time to this campus, and I appreciate you bringing in the great weather too, because everyone lies about what the weather normally is like here, right? So today I'm going to tell you about a study in the lab that's a joint effort by my lab and our neighboring lab, Xiaoyu Zhang. This was a little serendipitous how it occurred, but really this is a joint effort. He could be standing here giving you a very similar presentation. And so this is the work of a student in his lab, Bill Ricci, who's developed some of the technologies and implemented them from use in animals to plants. I'll highlight his contributions, as well as people from my lab. Zifu Liu helped adopt a TAC-seq for application to plant genomes to map chromatin accessibility, and then the people in my lab really led the data analysis. And this work is almost exclusively funded and supported by the National Science Foundation. And so one of the sort of long-term goals of our lab, it might be a bit lofty, but we really would like to find ways to induce variation, to induce allelic variation that we can lead to phenotypic variation that we could sort of introduce into breeding programs. And there's lots of efforts, especially on this campus, where you're using existing or standing genetic variation in those programs. But we want to look to the future and try to adopt these gene editing technologies in combination with what we're studying about, what we're learning about these plant genomes to sort of accelerate that process. And so I just use this as an example. Many of you are probably familiar with this work. There are other people that have done very similar experiments, but this is a nice example of what can result from inducing genetic variation at a non-coding regulatory element. So this is some nice work from Zach Lippman's lab, where they already knew that this gene CLAVATA3 is important for a whole range of developmental traits associated with tomato fruit shape and size. And what they did is they essentially carpet bombed this non-coding regulatory region within a couple kb upstream of the coding sequence. And from that, they were able to create a range of expression variation and also phenotypic variation associated with essentially expressing this gene or expressing it in different cell types possibly or different levels. And so this worked primarily because the locus that they're studying, most of the regulatory variation is actually found here within the neighboring 2 kilobase region of this locus. And that's actually true for a lot of plant genes. And for those of us, I'm guilty of this, that have worked on Arabidopsis most of our careers, that's where everything resides because that's the only place you can put it. But what I'll tell you today is by using chromatin structure, we can now identify rather acutely these, what we predict are regulatory elements in any plant genome. And so what are these regulatory elements? Your sort of classic textbook definition goes like this. You have a core, you have a gene, it has a core promoter where these general transcription factors assemble. And then you can have these regulatory elements that can appear either downstream or upstream of a gene, and they can occur in any orientation. And that's because in specific cell types, for example, here in the limb, there's a limb transcription factor as expressed, and it helps position and create this 3D conformation that helps provide cell type specific expression. And you can see here in this example, there are binding sites for brain transcription factors, but they're occluded by nucleosomes, and of course those transcription factors aren't present in these limb cells. So this is one area where I'm a little envious, actually was quite envious of people working and studying animal epigenomes, because for a long time now, almost a decade, if not longer, they have been able to take these very large genomes and use sort of functional epigenomics data and very quickly not only identify coding sequences and genes, but also identify these likely regulatory elements using a chromatin structure and other signatures. And so in animals, often what you find at these regions that are found out here, these distal regulatory elements, is that there are specific histone modifications that characterize these regions. They're enriched, as you might expect, for transcription factor binding. These regions have accessible chromatin that enables transcription factor binding, and part of that is because there's rapid nucleosome turnover often at these regions. Now, we could do many of these same experiments in plants. So, for example, plants also have H3K4 monomethylation. But in plants, this is not found in intergenic sequence. It's actually found in gene bodies. And so if we're just using this single histone modification to predict the location of these elements in plant genomes, it just didn't work. But what we hypothesized and what's expected is that there should be enrichment for transcription factor binding sites. And to enable that, these regions should be, have accessible chromatin. And so we worked hard a few years ago to try a whole range of experiments to adopt this method referred to as a TAC-seq for application to plant genomes. So this was developed by Will Greenleaf out of Stanford. And essentially the way this assay works is they can profile regions that essentially proteins can access in chromatin because they take advantage of a hyperactive transposase that will integrate DNA sequences in regions that are accessible compared to regions like here that are inaccessible. And so after doing that, there's a simple PCR reaction that reveals these accessible regions in the genome. And so you get these landscapes like you see here of these peaks that emerge. And what they could show quite nicely is that this method for mapping accessibility benchmarked really well to sort of standard accepted methodologies that were out there at the time. And what was nice about this particular assay is that the input for the number of cells was much lower than was in practice at the time. Of course, now this method is being used for single cells, right? Again, the challenge was applying this to plants. It's not hard to isolate nuclei. Many of us do it all the time. It's a simple incubation with this hyperactive transposase and PCR sequencing. And you can do this repetitively and spend a lot of money sequencing a lot of mitochondrial genome, right? And so actually, if you read a lot of the papers in the animal literature, you'll find a lot of the sequences are derived from the mitochondria. And of course, this doesn't have a typical chromatin state. It's highly accessible. It's acting as a sink for a lot of that transposase. And of course, in plants, the mitochondria is about 20 times larger. The genome is about 20 times larger than the human mitochondria. And then there are chloroplast genomes. And they're also large, and there are many chloroplasts often per cell. And so basically, in the lab, Lu, who finally got this to work, hypothesized that we just need to actually a cleaner way to isolate nuclei. So we've used this method where we couple nuclei sorting to a TAC-seq, and there's some nice work from Roger Deal where he's been using another method for nuclei isolation where then you can apply a TAC-seq. So what we've all learned as a community is you need pure nuclei, and you need to separate it from those organellar genomes. And this benchmarked really nicely to what was the gold standard accessibility data at the time in Arabidopsis, this DNA-seq data from Christine Quaich's lab. And at the time, we could show that we could, again, use this on 500 nuclei. Of course, it's, again, being used for single-cell nuclei now, so it's not that exciting. But it also worked on sorting nuclear localized GFP lines that were available. And so this is useful because there's a lot of pre-existing plant lines that are out there in the community because they study very specific cell types where they can sort, if they choose to, that specific cell line to profile that accessible chromatin environment. So the assay was working, and we turned to applying it to other genomes. And I won't go into the history of why we started doing this, but what I'll tell you is a light bulb turned on, and that's mostly because, and I'll show you how a TAC-seq helped us focus all these other epigenome data sets on really pinpointing these distal regulatory elements. So today I'm going to show you data, some RNA data. Again, it's mostly focused on looking at that TAC-seq data. We do have some whole genome bisulfite sequencing data to look at DNA methylation. Turns out this is another part that my lab studies. I'm not going to talk about it at all today, but it's kind of interesting that the regions that are accessible have essentially they're depleted for DNA methylation. So now I feel like I'm studying the entire genome. There's a whole variety of different chromatin modifications that we've also profiled. Don't worry about understanding and having to understand what each of these does. I'll point it out if it's important. And I'll show you later on how we've been using some more functional assays to really assign these regions to their target genes and then test for activity. And so this is sort of the landscape of the epigenome when you focus in on genes. And what I'll tell you is really nothing surprised us here. Everything that we already learned about the chromatin modification landscape at genes was predicted based on what we already knew from Arabidopsis. And turns out transcription in eukaryotes is a pretty highly conserved process. Well, of course, there are some variations. And so what you're seeing here is that the genes are ranked from their highest expressed gene to the lowest expressed gene. And the red line up here shows sort of a meta profile for the bottom 20% of expressed genes. And the blue line shows the meta profile for the top 20% of expressed genes. So you see a lot of high methylation here in the low expressed genes. And then another group of low expressed genes have H3K27 trimethylation. This is a modification imparted by the polycomb group proteins involved in repression. And then you can find other activating marks associated with activation. So K4 trimethylation, and you can see K36 trimethylation in gene bodies. So this just shows you, again, the data is of high quality, but again, everything that we observed here was what we had predicted based on our previous work in Arabidopsis. I'll show you how the data becomes really useful. We start trying to study the chromatin environment of these distal accessible chromatin regions. So, having worked on Arabidopsis for a large number of many years, this concept of distal regulation was foreign. Of course, it didn't exist, and there were of course well-known examples of maize, but those were nuances. They weren't really real, or they were just they became real really quickly. So here's one of them that was very real. This is the TB1 QTL in maize, and it's important because it's one of these major QTL that leads from this sort of bushy phenotype to this more upright stature, right? So it's important for inflorescence architecture. And many people, including a lot of work led by John Dobie's group, had really through brute force fine mapping identified about an 11 kb region that was 70 kb away from the coding sequence, the TB1 coding sequence. So it was a bona fide distal regulatory element, right, that was, again, cloned through this brute force mapping. And what they found was that if you look in that 11 kb region, that's the intergenic space, there is a region that has very low nucleotide diversity compared to nearby regions. It turns out this actually overlaps these transposable elements. And when they took the sequences underneath this region with very high sequence constraint and they fused it to a reporter, they could show that it can lead to activation of that reporter, right? Even showing functionally that these sequences, even though they're 70 kb away, can activate that reporter gene. So it's a really classic example of these distal, of the potential for distal regulation in plant genomes. And so when we generated our data, first place we looked at was sort of these handful of important loci that have been characterized over the years, really about 4 or 5 of them. And so this is the TB1 locus. So TB1, you can see, is located all the way over here. This is the coding sequence. Here's just a track of DNA methylation. Here's a track of H3K27 trimethylation. Just remember, again, this is involved in transcriptional repression. And then here's the ATAC-seq data. And this is what really opened our eyes. This is a really clean type of data that we're not used to working with such clean data. And you can find this really nice sharp peak. They're typically 200 to 400 base pairs in size of accessibility, right? And when we saw this, we were really excited. We thought, well, if only John Dobie's group had access to this while they're trying to map the sort of genetic basis for this QTL, this could have helped speed up this process. And so we dig back through the paper and we look at the region that they showed was important for activating that reporter. And then we were really disappointed because we found that this region is actually not in that QTL. And then we realized, because we're not developmental biologists, that TB1 isn't expressed in the leaves and you wouldn't predict it to be there. And so we repeated the experiment in the tissue that's important for TB1 control of inflorescence architecture, and that's in the young developing maize inflorescence. And so we did that experiment in the ear, and what we thought was quite remarkable. So this most distal region of accessibility is lost, and now you see these 2 new regions emerge, and they perfectly localized to this region that has been genetically characterized and functionally characterized to be important for sort of distally activating TB1. What you also see happen is the promoter now opens up, and now you can see an abundance of RNA transcripts coming from this gene, right? So this was exciting to us because, as I'll show you, this isn't just a nuanced example. It turns out these are really prevalent, not just in maize, but plants with large genomes. And so I'm going to refer to these as distal accessible regions. Actually, I'm going to almost primarily focus on these distal regions today. There's essentially a continuum, right? I'll show you examples where they start here over the transcriptional start site, and then again, they basically can get pushed out over time. We can speculate about how that might happen. But I want to focus on these distal ones because we've always sort of guessed as plant biologists, the sequences must be there within a couple kb. We'll take that, see if we can rescue or make reporters. But that was basically guesswork, right? But now we can pinpoint these regions, we think. This is not an isolated example, too. So here's another example of a recent QTL that was, again, cloned by John Dobley in collaboration with Feng Shan's lab, studying this flowering time adaptation locus. And what they found through GWAS was there's this region of really high linkage disequilibrium that occurred about 50 kilobases away from this gene CCT9, and this is where you have the strongest signal for that GWAS. And then we quickly looked in our data, and sure enough, right underneath that region, you find this region of accessibility. Again, I think, I really believe that these data are going to facilitate the identification of QTL, just like the annotation of genes does. So this is not rare, and this is no surprise to this crowd. There's been some really great work from Ed Buckler's group in collaboration with Hank Bass, where they have showed that even though these regions of chromatin accessibility are a minor fraction of the maize genome, they contain a tremendous amount of the heritable phenotypic variation underlying these complex traits. And so we really now are excited about further discovering these elements, not just in maize, but in other plant genomes. But I'll continue to focus on maize just for the first part of this talk. So we started to characterize these, right? What's their distribution to their nearest gene? And as you might expect, the majority of these regions are located with, very close to that initial, transcriptional start site. But if you look closely, you can see there's another bimodal peak. This other peak hits its peak at about 32 kilobases, right? This is, again, you think about the Arabidopsis genome, you can fit 8 or 9 genes in 32 kilobases, right? So this is, there are many of these, thousands of these. In fact, by our calculations, there's about a third of these accessible regions we consider distal, right? They're distant or far away from that gene. We can look at those intergenic accessible regions, and we can look at their GC content and find that it's very distinct from other nearby intergenic sequences, right? Which is important for nucleosome positioning and likely harboring these transcription factor binding sites. So they're distinct from other intergenic sequences. And then I won't go into details today, but we have a nice collaboration with Andrea Galabati's lab at Rutgers. And between our labs, we've been able to profile DNA-protein interactions for almost 50 transcription factors now in maize. And we can see when we look specifically at these distal regions, this is just showing enrichment this is the TSS. The same thing happens at these distal regions. You can find enrichment for binding. So what I'm trying to take you down a path where we can find these accessible regions, and I want to try to convince you that they're actually functional. And so this is part of that in that they can be bound by transcription factors. So if you talk to Ed and others, not the molecular biologists, this is function, right? You look at genetic diversity and evolutionary genetics. And so what we can do is we can take those accessible chromatin regions that exist that are distal, and we can look at their nucleotide diversity using sort of the maize diversity sequence data that's out there, and see that again at these regions of accessibility they're sequence constrained compared to nearby sequences. And we can take some other data from this GWAS catalog that Jason Maltz did while he was in Ed's lab and show that there's an enrichment for GWAS signal coming from these regions of distal chromatin accessibility. Again, indicating they're functional. And I think this is remarkable because this is just accessible data from leaf, right? It's a single tissue where these GWAS data come from a whole range of phenotypes. So I think this signal gets stronger and stronger as we resolve the number of cell types that we can profile accessible chromatin from. And then in a really nice collaboration, with an ongoing collaboration with Catherine May-Guerra, who's here in the audience, we used the data from the recent EQTL study by Ed's lab, and we could show that there's a really strong enrichment for these EQTL, again, in these distal accessible chromatin region. Again, indicating these regions are functionally important, all right? So that's the evolutionary genetics. Now, I told you earlier, when we use chromatin modification data, we didn't really learn anything when we looked at gene space. But we started to characterize these distal chromatin accessible regions based on their local chromatin modification environment. And I think it becomes quite revealing and predictive likely of what's happening at these regions. So all of we basically found there's 4 different chromatin environments. And so all of these next 4 classes I'm going to show you, they all have accessibility. So this is just showing accessibility. Again, these are distal accessible regions. So they're far away from nearby genes. So this particular class has acetylation. So it doesn't matter which type of acetylation we look at, you see enrichment of histone acetylation flanking, this is important, flanking both sides of the chromatin accessible region. Not one side, both sides. Right? And so you would predict then these will be involved in transcriptional activation. Right? The other class we find are these ones that are flanked by H3K27 trimethylation. So you see that here. Again, they're accessible. There's H3K27 trimethylation, and the other modifications are depleted. And there's a lot of great literature. We understand very well about how this modification and its function in sort of repressing gene activity. So the prediction is these are distal regions that are imparting repression on their target genes. The next group we found threw us a bit for a loop, because if you look at it, it looks just like a transcribed gene, and that's why we call these a transcribed group, right? There is basically enrichment for K4 trimethylation. There's H3K36 trimethylation in the body of that region, and you see RNAs actually coming from these regions. And they look like genes because we actually think they're genes. They're unannotated genes in this reference genome, or they're unannotated long non-coding RNAs. Right? So even though we found them as distal accessible region because using the annotation they were far away from the nearest gene, we just think that they're not annotated. So I'm not going to talk about these any further. It's not to, obviously if an unannotated gene it's not that interesting, but it's not to minimize that some of these could be important for control of nearby genes if they're long non-coding RNAs, but these aren't sort of this classic, like the first two groups I showed you. We think this is a bit of an annotation artifact. And the last group we found were just completely unmodified. So there's plenty, these are accessible regions. Again, they're distal from genes, and there's no modification around them. And these are actually the most abundant of all the classes that we've identified. So when you look at these 4 genes, we can, again, start to try to functionally characterize them using gene ontology. And what you see is, again, we're playing a little bit of a guessing game, because we know there's a region of accessibility, but we don't know if it's associating with the gene on the left or the right, or if it skips a gene. So what we did is we took genes on both sides. We used this in gene ontology, and what we found for the acetylated group are terms of what you would respect, right? These are response stimuli consistent with activation. The K27 trimethylation group and this unmodified group, all these terms associated with transcription and regulation, DNA binding, right? This is, again, what we'd expect. A lot of transcription factors in plants are controlled in a very specific manner by silencing, either silencing of K27 or removal of K27 trimethylation. And then, like I told you earlier, this transcribed group, which I'm not going to focus on so much, when we use GO, it actually come back with no terms. I didn't think that was possible to get no terms, but you can get no terms from GO. So where we've struggled is actually with then taking these ACRs, because we have these predictions. The acetylated should be associated with transcribed genes. The K27 should be associated with repressed genes. And we try every which way. We never could find these clear associations with their nearest genes. Again, we're a little bit of a guessing game. We don't know if it's the left or right gene. And so I'll come back to this point later on. But this basically reveals 2 major challenges that we faced at the time. One is we need to find a way to associate these distal accessible chromatin regions with their target gene, right? And then the other is just because they're accessible doesn't mean they're functional, right? So the way we got at assigning these distal accessible chromatin regions to their nearby gene was using these proximity ligation methods. So you might have heard of Hi-C. We're using a variant called Hi-CHIP, and I'll explain how those work. And then I'll show you in a few more slides how we've been able to functionally demonstrate at least some of these accessible regions have the capacity to activate a reporter gene using this method called star-seq. So typically what we do in the plant community, actually many of us, in regardless of what we work on, you think you find an important regulatory element, you stick it upstream of a reporter, and you ask, can it activate that reporter expression? So I'm going to show you a method that we've adopted again from, we first developed for use in Drosophila, and how we've applied that to the maize genome. So assigning genes in these distal accessible chromatin regions to their target genes, we're all guilty of looking at genome browsers and thinking the genome's this really long two-dimensional space, but we know it's highly compartmentalized and organized in the nucleus. And so you can, looking at genome browser, these are supposed to be nucleosomes. You can find regions of accessibility, and then maybe there's a region of distal accessibility over here. But in reality, what happens in three-dimensional space is there's a loop that's forming here, right? And this can be mediated in part by sequence-specific factors, among others. And so you can map these sort of three-dimensional interactions using proximity ligation methods like Hi-C. And so what you can do is you can freeze, essentially in time, this chromatin sort of compartmentalization. You digest the genome and you religate, and sometimes you religate sequences that were distal or basically far apart in two-dimensional space, but it tells you they must have been close together in three-dimensional space. So for example, we can map maybe the interactions that occur here that really are in two-dimensional space very far away. Now the challenge with Hi-C is you need really deep pockets. especially for, the maze genome is not that different from the human or mouse genome, but something that really has, I think, plagued the plant community for a long time, which is changing rapidly, will change very rapidly, is we're always mixing cell types, right? We're taking like a whole leaf. I always say it's like chopping off your arm and grinding it up. It's like there's so much complexity in these tissues, above ground aerial parts you'll see in methods, right? And so you're not just doing high C for a cell line, your signal is completely diluted by all those different sort of high C patterns that would emerge in different cell types. So this just really wasn't something that we could do. And I say we couldn't do it. Of course, we did it, and then we realized we shouldn't do it. So I'll show you the next slide how that works. But what we realized is because the amount of sequencing that would have been required to really do this experiment the right way, that we used a modified version. Again, we're really fortunate because there was some nice methodology where we used a variant called HiChip from Howard Chang and again, Will Greenleaf's lab from Stanford University, which basically allows you to capture these chromatin conformations, right, using proximity ligation, and then you follow up this experiment with immunoprecipitation. So we're using antibodies against, you can use against transcription factors, we're using antibodies against, again, specific histone modifications. So using this method, we're only finding interactions that occur where these histone modifications are found in the genome. And if you remember, many of these regions that are accessible and distal to genes, they have specific chromatin modification environment around them. So this allows us to try to test what genes they interact with. Now, this doesn't resolve the cell type issue, right? But what it does allow us to do, somewhat superficially, is at least separate the transcribed active genes from the repressed genes. So maybe it simplifies that state to the expressed state and the repressed state is one idea. And because of that, because of separating expression states and then sort of the trimmed down version, not looking at all pairwise interactions in the genome, require many fewer sequencing reads. So this becomes achievable to test unlimited sample numbers. So this is what our high C data looked like at the time. Again, this is a contact probability map. And so what you're looking at is this blue is this background color. And here's the TB1 gene when this arrow is located. And then what you see here is that distal accessible region that's 70 kb away. And what you can generally see, anytime you see heat on this map, what you're basically seeing is that there's basically a large interaction occurring here between this whole domain. It's basically folding up on itself. But when we, again, separated this domain based on the repressed allele versus what could be seen as the active allele, you see a very different picture when we use HiChip, right? So now this becomes very apparent. And this big X you see here is just a region where we can't map the DNA. It's a highly repetitive genome, so that's why it looks like it's background. But you can say very clearly that if you wanted to try to assign this distal accessible region to the gene over here, it's very clear it's actually interacting with the TB1 gene, right? So this is allowing us now to assign these genes or these accessible regions to their target genes. And I'd like to show this not in one example. I should preface this. We actually don't know if this is causal, but it's being tested. But it's a nice example because people like Sarah Hake and Dave Jackson that have worked on this for many years, among others, they'll tell you we've never been able to rescue the mutant phenotype using endogenous sequences, right? We're missing something. And Dave's like, we've even used 50 kilobases upstream, doesn't work. And so what you look is, if you look at the knotted 1 locus, again, HiChip, you kind of squint your eyes and see something. With high C, you squint your eyes. With HiChip, it's so clear. So again, here's the knotted 1 coding sequence. And here's this distal accessible region. It's 91 kb away from knotted 1. And you see this beautiful loop that's forming here, right? It's just perfectly interacting with the knotted 1 locus. And so now, what people in that field can do is they can go in here and use loss of function studies. Right? This becomes a candidate for, we talked about earlier, for targeting with CRISPR mutagenesis, right? Edit this region, see if you can then create that mutant phenotype or modulate that mutant phenotype. Secondly, you can do gain-of-function assays. You can take these sequences out here and start to basically move them next to these, the endogenous gene to see if you can actually rescue that phenotype. So this is how we think these data will be useful. So Bill Ricci in the lab, he's the one that got HiChip up and running, and he started looking through the analysis. And in any of these sort of, anytime you're running genome-wide analysis, you're basically looking at confidence, right? You're looking at confidence for interaction, for peak calling. And what Bill noticed, I thought, was actually quite remarkable. He discovered that what we already knew is that genes themselves end up being hubs for lots of chromatin interactions. So he puts these anchors here because you see a lot of interactions extending from these genes. But if you factor in the strength of the interaction, right? This is based on a statistical test. And the strength here is increasing as you go down here. What he notices, that strength of interaction was highly predictive for what are likely sequence-specific loop interactions. So here you have the TB1 region that you've seen a few times. Here's a TB1 coding sequence. There are multiple interactions that occur with TB1, but notice this one here with basically the strongest p-value. It perfectly localizes to that region that's known to associate with the TB1 locus. And he could show this, again, in collaboration with Catherine and Ed's lab using their EQTL data, again, looking at some of the strongest, looking at some of the EQTL. When you rank these chromatin interactions based on their p-value, it's very, very indicative, likely, of sequence-specific interactions versus sort of localized chromatin interactions that occur just because they're in the similar neighborhood. So Bill could go on and show, especially for the K4 trimethylation data, this was especially true. Overall, there's a significant proportion of these EQTL distal accessible region gene pairs that are highly enriched. And when you factor in this strength of interaction, look at the EQTL loops compared to all the loops, right? You factor in, look at their p-values. They're really strong, right? So I think this is really important, and we haven't seen this before in any species where you factor in the strength of this interaction that actually could be predictive for a true sort of sequence-specific interaction versus these sort of localized chromatin contacts that can happen. And remember earlier, I told you that when we try to associate, it's predictive. These regions of chromatin accessibility with histone acetylation, you would predict will be associated with a transcribed gene, and vice versa for K27 tri, and that never worked for us. And again, we could even use our loop data here, and we know what gene it associates with now, and it's still really noisy. But when we factor in, again, the strength of the interaction here, using the high chip data, now it becomes much clearer, right? You see that the K27 regions are very lowly expressed, and the K acetylation regions are very highly expressed. And so it finally started to resolve those expectations that we had from the beginning, but it only happened when we factored in the strength of these interactions. And so basically what you see happening is, and this isn't to offend any of the Maze researchers here in the audience, but what it feels like and looks like is probably happening is the Maze genome has essentially transformed itself into the Arabidopsis genome, right? So if you look at the DNA methylation tract, what you see here is all this methylation, and what you'll notice is when regions are unmethylated, you either have a transcribed unit there or you have these accessible chromatin regions. And then what you're seeing here are, in the black boxes, are edges of loops, right? And you can see those loops are highly enriched in these regions that are not DNA methylated, right? And so what Bill has hypothesized, which I think he's spot on, is that what you're seeing happening here is you're looping out all this sort of large domains of H3K9 methylation and DNA methylation that doesn't contain any regulatory information. And you're sort of transforming that large Maze genome into this little trim Arabidopsis genome here, just as an analogy. And so you can now place those sequence elements nearby their target genes. And so in the high chip data, what you often see is, again, these sort of localized chromatin interactions. And they're real, but do they just happen because they're nearby each other now, right? Versus what we hypothesize when you factor in strength of that interaction, we hypothesize that these are mediated by sequence-specific factors, right? Likely transcription factors, and there are likely others. There's no CTCF in plants, but we're thinking along these lines. Okay, so we can identify these distal accessible chromatin regions. We can, with high confidence, to a subset of them, assign them to their target genes, but are they functional? And one way we've tried to get at that is to adopt, again, this methodology that was first sort of applied to Drosophila to plant genomes. And so this is a method called StarSeq, developed by Alex Stark's group, and what they learned was they basically took advantage of the gene model, right? We think of this as enhancer, promoter, expression. And of course, as I've showed you throughout this presentation, we know that these enhancers can occur downstream and then through 3D conformation loop around and interact with that core promoter to drive expression. So they took advantage of this principle, and what they did was they chopped up the entire Drosophila genome into 200 to 300 base pair fragments, and they cloned it into this system, where instead of now having a node enhancer here, or one you're testing, you're just testing all possible fragments in the Drosophila genome. You take this library of clones that you've created, you transfect that into these Drosophila S2 cells, you incubate, give it time to induce transcription, and anytime a sequence in here is able to interact with the core promoter to drive expression, it ends up transcribing itself, And then you can specifically capture these mRNAs that come from this vector. You sequence the RNA, and you can at least find elements that can drive expression, right? So it won't be useful for finding those that can lead to repression, but it can find those that can activate expression in the system. So again, this is, again, work from Bill, where he worked almost 2 years on this, and it's working like a charm now. So what he did was he took this library of ATAC-seq fragments. So I'm going to show you two slides, two different results in the next slide. One of them is a pilot where we just took the backclone where the TB1 locus resides. Because of the costs involved here, we could chop that up into smaller fragments and test it. And then what we did was, because we couldn't afford, we didn't think at the time, afford to do the whole maize transcriptome, the whole maize genome, we just hypothesized that, well, we're testing these accessible regions for activity. So we devised a way to clone in an ATAC-seq library into the star vector. So we were trying to test those specific regions. It turns out we tested pretty much the whole genome. But so, because there's always background in any library you make, and so we were able to see that. So we have a very similar setup. There's a minimal 35S promoter. There's a small open reading frame spacer again. We clone in the fragments here. It has this poly A tail that allows you to capture it. And Bill spent hours and many nights growing liters and liters of E. coli to generate a really diverse library that he then had to get into efficiently into maize protoplast. And he was able, this is just a control showing GFP, but he was able to achieve over 60% transformation efficiency of these maize protoplasts. And from that, we're able to survey essentially 15 million independent events. And what's great about this assay is there's not cost really for sequencing. The sequencing is cheap. The cost is in the production of all these libraries. And so at the end, what you do is you sequence RNA and DNA, and you compare how much RNA do you recover versus how much input DNA did you start with. And that's a measure of activity. And so again, here's the experiment we did in the back clone. So this is, again, the TB1 locus. Here's the coding sequence. Here are these distal accessible regions. Here's one we think is a repressor. This is one we think is involved in activation. And what you see very nicely is that there's this background sort of DNA level you recover. And then you see activity. And you can see here, there's reporter activity coming perfectly from this region that John Dolby's group showed could activate that reporter, right? You also see activity coming from that sort of promoter region as well. So this assay is working. We can apply this genome-wide. And what we see is we basically, again, separated those distal accessible regions based on their chromatin environment. So I know I kind of sort of tossed this group aside earlier, but it's a little convenient now because even though we don't think they're distal accessible regions, they're genes. And what you see is this is a ratio of DNA to RNA. So anything that pops off on the x-axis is showing transcriptional activity. And so what you see for the transcribed group is these elements reported the most activity, which again, you might predict because they're actually transcribed genes. But what also happens is you see this happen at these distal accessible regions. Now these are not genes, these are distal accessible regions. Many of these show reporter activity. And then the unmodified and the K27 group have very few, there's very low evidence that they show activity, and this actually fits with their proposed functions. And so we think we've shown, at least for a subset of these regions, that they actually can lead to transcriptional activation. Now keep in mind, anytime you do an assay like this, you're testing the interaction of that sequence with that minimal promoter. And so we have a nice collaboration with Christina Kweitjes where they're trying to innovate and develop a whole range of novel minimal promoters to basically sample different elements that can interact with those promoters to drive expression. Okay. Again, what's important, just to come back, when you think about that sort of definition of what are these regulatory elements, they can be sort of upstream, downstream, any orientation. What we didn't realize at the time that was pointed out to us was, the way we cloned in this library is unbiased. It goes in both ways. And so we were able to test of all those regions that showed activation, did it matter the orientation of the insert? Turns out it doesn't. Over 90% of these regions showed activity from both orientations, right? And it didn't matter whether or not they were upstream or downstream of their target gene. So we really believe these are bona fide sort of enhancer sequences. Okay, so I'm going to stop talking about maize and just transition to some of the comparative work then that we've started to do in looking at other plant species. And maize really was the one we drilled down deep on, and now we're trying to go wide in many other grass species, but we also try to be representative. Of course, I say representative, but most of our genomes are from crops because it's already a biased group. But we performed a TAC-seq in all these other species. We try to focus on leaf sample to keep it somewhat similar. I appreciate there are really differences in biology between leaves of different species. But I think there's some clear take-home points here. So one of them is you can see that the total amount of accessible space that's here in these genomes is actually quite similar, regardless of genome size. So what you're seeing here is these species are ordered based on their genome size. So Arabidopsis has about 100 megabase genome. Barley has about a 5 gig base genome. And regardless of the genome size, the amount of sequence underlying accessible chromatin is about the same. And if you plot that as a proportion of the genome, of course, what you see is the bigger the genome, the lower proportion of the genome they represent. And what I think this is communicating to us is that these are limited in the genome. They're not proliferating, right? They're just like genes in many ways. Because they're associated with genes. Again, I showed you earlier in maize that there's really this bimodality of their location to their nearest gene. They're either right over the transcriptional start site, or you start to see this other peak that occurs further away from the gene. And when we rank, again, these genes based on their genome size, you see, for example, duckweed and Arabidopsis, most of their regions are right next to the transcriptional start site, and some of them get to a kb away. But when you look at these bigger genomes like asparagus and barley and maize, you see many more further away. It correlates very highly with the size of the genome. With bigger genomes, these regions can be found further away from likely their target genes. And you can see that based on this classification system we use. Do they overlap the gene? Are they proximal to the gene? Or are they distal? You can see that, again, with increasing genome size, they just move further and further away from the gene. And we can all speculate later about how that happens, but we can talk about it if there's interest. So we also performed the same chip experiments looking at sort of key chromatin modifications in all of these species. And just like before, we have this transcribed group. So this transcribed group in the top row has K4 trimethylation, histone acetylation, and body K36. That's what a transcribed locus looks like. So no surprise here, the accessible regions that are over the TSS have those chromatin modifications. But again, as you look at the distance, so these become proximal, it's moving further away from the gene, and now you get to these distal regions, right? They're far away from the gene. What you see happen is you lose association with chromatin modifications that are associated with transcription, and what you again find are these three main groups. You find this unmodified group, which by far and away is the largest classification in all of them, and then you find this K27 trimethylation and histone acetylation group. So again, this is not a feature of the maize genome. This is a feature of flowering plant genomes. It's consistent. Again, we can take gene ontology like we did for maize, and what happens is whenever you look, there's a lot going on here, but long story short is, if you look at that K27 group and that unmodified group, in every species we look at, when you do gene ontology, you always find DNA binding and regulation. Right? Again, we think these are critical regions that are distal regulating key transcription factors important for development or response to the environment. Again, just like in maize, we can look at all the sequence diversity panels that are out there for all of these species. We can look at these distal accessible chromatin regions, and what we can see is, just like in maize, they're depleted for sequence variation compared to nearby sequences. Again, indicating some level of sequence constraint. And then we can try, this is where we fell miserably because we're not very good evolutionary geneticists, but we try. But, we can take advantage, for example, of these 5 related grasses, right? And you can see the region of synteny here, and there's this region of accessibility that's shared amongst all of them, and that red box is showing you a sequence that's highly conserved, right? So you can start to think about the sort of the role of conserved non-coding sequences that help drive these accessible chromatin regions. And what we can see is that when we look at these accessible chromatin regions, so we identify conserved non-coding sequences between all of these, all the dicots, and then separately all the monocots. And what we can see very, very nicely is they're highly enriched for these conserved non-coding sequences, right, in these distal accessible chromatin regions. And again, when you look at the chromatin environment, these 4 different chromatin classes here, you can see that even between distantly related species, these CNSs have these conserved chromatin modifications. So here's a class that all have K27 between all species. Right? Here's a class that's transcribed and so forth. So there are sequence determinants that are leading to them being accessible, but they're highly conserved in some instances. Here's an example I like to show because I think it starts to help you think about how the landscape of chromatin accessibility can form in these species. So again, this is a region of synteny between maize and sorghum. And what you can see here is there's a region of accessibility at basically right at the transcriptional start site for the sorghum locus. If you take the sequence underneath this and blast it to the maize genome, you get one hit, and it maps perfectly to this accessible chromatin region that's here, that's now 100 kb away from its likely target gene, right? So you can start to think, what happened, right? Did a transposon sort of snuggle in there and just create a space? And then, of course, over time, it's easier to push it further away. That's one idea. And then, of course, it's very hard to have such acute deletions that delete just this region but not this accessible chromatin region. So you can start to think about how these evolve. But, of course, another way in which that can happen is, well, the way in which you push them away is because there's transposon content, right? So we compare these instances between maize and sorghum. What we find is the distance between themselves and their target gene is highly correlated with the transposon content. Really not a surprise given that's what's in this intergenic space, but it helps you think about how transposons can shape the accessibility landscape in genomes. And at the same time, we can start to think about that. I always forget to say this. Both the TB1 example and the CCT9 example are accessible regions that are embedded in transposons, right? And so there are examples where transposons can harbor these regions. They can create new regulation. And so transposon biologists sometimes don't like to show. If I show this to Nathan Springer and say, oh, if we look on average, right, accessible regions that are fully embedded within transposons are depleted in the genome. But he'll say, but they're there. And he's right, right? We wouldn't expect every transposon to have accessible chromatin, and they clearly don't because they're depleted compared to what you expect by random chance, but they are there. And when we categorize all these different species based on their transposon content, what we find is that certain classes of these DNA transposons are highly enriched in these accessible chromatin regions, and as Cedric and others would tell you, it's a bit of a circular argument when you're looking at these DNA transposons because, remember, we're using a transposase to actually define these regions. But you'll also find certain classes of retros also that can have accessible regions. So we're trying to focus in on this more in collaboration with Nathan Springer. So I'm just going to wrap up. I hope I've convinced you that there are, and distal regulation in plant genomes actually is occur. It's not a nuance. I think it's going to be far more common, especially think about all the existing QTL studies that are out there. I really think, and GWAS experiments, I think this will help facilitate the identification of the causal variants, if that's of interest. We can use chromatin modification and accessibility to uncover and make predictions about how that element might interact with the gene. And again, we can use chromatin contact interactions and the strength of that interaction to maybe predict, again, the sequence-specific interactions versus just localized chromatin interactions that occur because you're in similar subdomains of the chromatin. And what we're doing now is we're basically continuing this path of creating maps across development and time and species to really characterize and help create a compendium, really, of these sort of putative regulatory elements in plant genomes. And we're trying to follow that with some more functional and some variations of star-seq that we're trying to method. We've been doing some single-cell attack-seq, which is working really nicely. And, of course, what we'd like to do, getting back to that first slide, is start functionally testing these, right? So this is a little bit harder for my lab because we don't, there aren't many in Arabidopsis, and they're not the ones that are interesting to study, but we talk about creating allelic variation again at these regions. If they're associated with genes with no function, it'd be fun to start modifying them, which we're trying to do, to see if you can induce that variation again rapidly. All right, so I'm going to wrap up again. Thank the great work from Bill, and again, this really strong collaboration I have with a neighboring lab, with Xiaoyu Zhang. Again, he could be presenting the same data to you. And then the people in my lab that participated in again, the food developed the attack-seq methodology for application to plants, and then all three of the people in my lab drove the data analysis. And again, we've had a lot of support from collaborators, some of them that are here. And again, none of this is possible without the support of NSF and the Plant Genome Research Program. And with that, I'll stop and thank you for your time and take any questions if you have any.
I'll have you call on people and then repeat the question so that they'll hear through the mic. I've asked Bob to go ahead and mediate the questions here. So raise your hand when you're ready with a question, and he'll call on you.
Yeah, Paul.
So I have a question about these 4 categories of peaks, the ones that have histone acetylation, K27 methylation, transcribed or unmodified histones. I'm wondering if they have any differences in conservation among those categories when you compare them to other species. and also whether there are any transcription factor motifs that are enriched with those that might also be conserved or not.
Yeah, so the question is, when we look at those categories of four the categories are these distal accessible chromatin regions based on their local chromatin environment, how conserved are they, or are certain classes more conserved than others, and are they enriched for transcription factor binding motif? So I'll answer the second part first, because the transcription factor binding motif is interesting. So we can see that, at least in maize, these regions are highly enriched in transcription factor binding motifs. But there are some in plants, polycomb, in many cases, is recruited to targets based on interactions with transcription factors. And there's some well-known examples that maybe are core recruiters. And so we look specifically at those, and we can find some of those motifs underlying those regions, right? So we think, again, a major mode for polycomb repression in plants is actually TF recruitment of polycomb based on those elements. To look at the conservation, this is hard, right? So even though I showed you these conserved non-coding sequences, we've taken thousands and thousands of elements, and we're down to about 500, right? So the numbers become really low really quickly. And I'll tell you, and I know there's some experts here, this is not something our lab is really good at. We're using pretty primitive sequence alignment methods, and it's any you that studied intergenic sequence, it turns over fast. And so we can see, all the time, we can see these synstenic regions. There's clear regions of accessibility upstream both, and you look at the sequence underneath them, and it's, to take a 6 base pair motif to maintain it. And so this is where we've really struggled. Maybe Catherine has ways to solve that problem. Yeah, Cedric.
I have 3 questions.
You get 1.
3 words, no comment. You see what I'm doing. 1. Mappability. So you're still missing a lot of stuff because a lot of genomes are highly repetitive. So how much are you missing? 1. 2. Redundancy. So in mammals, mammalian genomes, a common theme with an answer is very rapid turnover, over evolutionary. So in multiple answers for given gene. You see how many? Sorry, I forgot that answers, but these ACRs, right? How many ACRs per gene? And number 3, unmodified. The unmodified, they don't, what do you think they are? And they are the most common, but they don't seem to be cis-regulatory, at least in your assay. They are starting. What are they then? That's all.
Okay.
Great job.
Thanks. So Cedric had 3 questions. One is, how does mappability affect the analysis? And it's a great question. It's something that you just can't avoid, right? Especially in a highly repetitive genome. I will tell you that we always map the data 2 ways. We always do sort of unique, sort of very conservative mapping, and that's typically what we use in publication. But we always allow reads to map everywhere too, especially for things like the element at TB1 I didn't show you. It's actually found in 3 different locations. In general, I don't think that big of an issue, because repetitive regions are typically silenced and heterochromatinized very quickly in plant genomes. And so these regions of accessibility are not found in those regions. They're anti-correlated. Not to trivialize it, I'm sure we missed some, but I don't think it's a major concern. This may give you a heart attack, but we actually use paired end 35 base pair reads because we don't pick up many more reads when we use paired end 150, right? So we're always trying to save money and push the number of samples we can use. The second question was how often these turn over. Again, this is not something we've really addressed yet. When we start to use these, we're talking about 12 million years of evolution, it's still far apart, right? And so we can always find examples, but we get to hundreds, sometimes dozens between species. So we don't ever make too many inferences about what's happening because we don't think we have the power to do that.
But for given tissue like this, on average,
how many
do you find per gene?
Yeah, so. No. So if the question is how many accessible chromatin regions are there per gene, and I would say on average 1, right? 1 to 2, right? The TB1 example is not common, where in 2 different tissues you see 3 plus the promoter 4 regions, right? That's not that common, right? And if you look at the numbers, these K27 group and the K-acetylation group, you're talking about 1,000, right? And I didn't show you the data, but if you look between different tissues, for example, root or ear and leaf, about 13 or 14% are differential. Right? So, again, this will, differential, we're grinding up really complex tissues, but I think this will change with a single cell data. And the unmodified group, we don't know, but the GO terms are quite intriguing, right? And so one thing we have shown, it's never all or none, but when we look at those different tissues and we see in one tissue it's unmodified, and then all of a sudden in ear, does it become modified, right? We can see evidence of that. So it's unmodified at that time, but again, we're looking at really limited numbers.
But it's star-seq. Okay, so you're doing this in photoblast, right? So they may not be active there, is that what you're saying?
Yeah, and there's lots of caveats with our star-seq. I mean, I think it's amazing, but there are caveats, right? I mean, I'm showing you the TV1 example, but I showed you TV1 is not expressed in leaves, and well, we're losing leaf protoplasts. Why is it active? Well, you've taken something, put it on a plasmid, you've removed it from its native chromatin environment. We can see that over and over in our data. So, I mean, it's great. It helps push the field forward, but there are some biological limitations of the assay. So I think there are many more as well.
Thank you.
Yeah.
What was probably the last one?
And John.
So just to follow up on the low number of ACRs that you're seeing for gene, I mean, you're quantifying those both by just looking to see what's nearby, but also by high chip. Or how do you come up with those numbers, and how far are we reaching?
Yeah,
It
so
seems like a low number relative to the 10 or 12 or so that we see.
yeah, we do not see anything like that, right? And so again, I don't know how much of it is because there are peaks that are being drowned out because, I used to do a lot of work on bisulfite sequencing, but we're looking at a dominant cell type when you have mixed cell populations. So that might be resolved from looking at single cell ataxic data. But we haven't really incorporated the high chip data to test for that. I will tell you from our high chip data, what we can see is that actually making the guess that the accessible regions upstream is a pretty good guess. 80% of the time, based on high chip data, that's right. I can tell you that's how we use the data. But yeah, the number, at least at this point. So we've done some environmental treatments as well, and really early on, and then we realized, I always call this the squinty eye test in lab. We're looking at the data and we're like, oh, there's kind of something there. But again, I think it's this issue of it's happening in some cell types, not others, and then we put it on the shelves and focused on function. But I think we're getting back to those now, especially again with how well a single cell ataxia works in plants as well. It's early days, I think.
Okay, thanks very much, Bob. Thanks a lot for the great talk.
This has been a production of Cornell University, on the web at cornell.edu.